Welcome to Nexus Clinical. Before diving into this lecture, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures. Biologic therapeutics represent a cornerstone of modern medicine, yet their large size and molecular complexity introduce significant challenges related to stability and manufacturing consistency. Unlike small molecule drugs, biologics are inherently fragile, susceptible to a range of physical and chemical degradation pathways that can compromise their safety and efficacy. This report provides a comprehensive analysis of the strategies required to manage these stability challenges, from initial process development through to final patient administration. It details the primary degradation pathways, including aggregation, denaturation, oxidation, and deamidation, and establishes the central role of critical quality attributes within a quality by design framework. By defining and controlling critical quality attributes, manufacturers can create a robust control strategy that ensures consistent product quality. This module correlates key critical quality attributes, such as aggregation, glycosylation patterns, and charge variants, directly to their potential impact on clinical performance, including immunogenicity, pharmacokinetics, and potency. Furthermore, it addresses the complex operational hurdles of sterile compounding and cold chain logistics, highlighting best practices to maintain product integrity up to the point of patient care. The central finding is that ensuring batch-to-batch clinical consistency requires a holistic, life cycle-wide control strategy. This strategy must be built upon a deep scientific understanding of how process parameters and material attributes affect critical quality attributes, and how variations in these attributes ultimately translate to clinical outcomes. Achieving mastery in the handling and control of biologic therapeutics requires a deep understanding of their inherent instability and the comprehensive quality frameworks used to manage them. Key learning objectives include defining and differentiating biologic complexity by understanding the physicochemical properties that distinguish therapeutic proteins, such as molecular size and complex three-dimensional conformation, from traditional small molecule drugs. These characteristics contribute to the inherent fragility of biologics. It is essential to identify the degradation pathways that compromise biologic integrity. This involves differentiating between the primary pathways of physical instability, such as aggregation, denaturation, and surface adsorption, and chemical degradation processes like oxidation, deamidation, and hydrolysis. A thorough understanding of the quality by design framework is crucial. This includes explaining the concept of critical quality attributes and describing how these attributes are identified and prioritized within the framework. The process leverages the product profile and risk assessments to ensure quality. It is important to recognize the relationship between aggregation and safety. Specifically, protein aggregates and subvisible particles can increase immunogenicity, which may lead to the formation of anti-drug antibodies. These immune responses can have significant clinical consequences. Analyzing the impact of glycosylation variations, such as afucosylation and sialylation, can reveal their effects on the efficacy of monoclonal antibodies. Variations influence effector functions, including antibody-dependent cellular cytotoxicity, and affect pharmacokinetics, including serum half-life and clearance. Applying analytical methods is fundamental for monitoring and quantifying critical quality attributes. For example, techniques such as size exclusion chromatography Ion exchange chromatography, and peptide mapping are used to analyze size variants, charge variants, and post-translational modifications. Describing the control strategy involves outlining components of an enhanced manufacturing control system. This includes the role of process analytical technology and the scientific basis for operational flexibility through the establishment of design spaces. Evaluating formulation components requires understanding the roles of buffers, ionic strength agents, and excipients such as surfactants and sugars. These elements help create a microenvironment that stabilizes the protein against anticipated stresses. Applying standards for aseptic compounding is vital. Relevant guidelines include United States Pharmacopeia standards, which focus on the risks during sterile preparations, such as incompatibility of diluents, like dextrose causing aggregation and adsorption to contact materials. Finally, assessing lifecycle management involves summarizing the requirements for maintaining an unbroken, temperature-controlled supply chain, often referred to as the cold chain logistics. It also includes understanding the regulatory importance of comparability exercises during manufacturing changes to ensure product consistency and safety. The development and handling of biologic drugs are primarily governed by their inherent fragility. Their large, complex structures are held together by a fragile balance of forces, making them highly sensitive to environmental changes. Understanding their physicochemical properties and main degradation pathways is the first step in creating a robust strategy to maintain product quality, safety, and efficacy throughout its life cycle. Therapeutic proteins possess unique characteristics that distinguish them from traditional small molecule pharmaceuticals. Their molecular complexity is remarkable. A large biologic drug can consist of more than 1,300 amino acids and have a molecular weight of 150,000 grams per mole. This contrasts sharply with a small molecule like aspirin, which contains only 21 atoms. Such complexity results in a precise three-dimensional structure or conformation, which is crucial for the drug's biological activity and efficacy. However, this intricate structure is only marginally stable. Therapeutic proteins remain stable only within a very narrow range of environmental conditions, including concentration, temperature, ionic strength, and pH levels. Their conformation is maintained by a combination of relatively weak, non-covalent forces such as van der Waals forces, hydrogen bonds, and hydrophobic interactions. These forces are easily disrupted, making stability a significant concern. Key physicochemical properties, including solubility, lipophilicity, the dissociation constant, and polarity, play vital roles in determining the protein's stability. These properties also influence how the protein interacts with biological systems, affecting its absorption, distribution, metabolism, and excretion. Understanding these properties early in the development process is essential for optimizing the drug's stability and predicting its pharmacokinetic behavior. Physical degradation pathways involve changes to the higher-order structure or multi-unit association of the protein without altering its covalent chemical structure. These pathways are a primary concern for product quality. One major degradation pathway is aggregation, which is the self-association of protein molecules. This process is a significant concern for manufacturers of biopharmaceutical products. Aggregation can occur when the delicate balance of forces that maintain the native structure of the protein is disrupted. When this happens, internal hydrophobic regions of the protein are exposed. These exposed areas can then interact with similar regions on other protein molecules, leading to the formation of larger complexes that are often insoluble. Aggregation can be classified as either reversible or irreversible. Reversible aggregation, also known as native aggregation, involves the maintenance of the protein structure, allowing the aggregates to dissociate back into individual molecules. Irreversible aggregation, on the other hand, involves denaturation and permanent structural changes to the protein. Several factors can trigger aggregation. Fluctuations in temperature and pH can destabilize the protein. Mechanical stresses resulting from agitation or pumping can also promote aggregation. Damaging freeze-thaw cycles can disrupt protein stability, and interactions with surfaces or contaminants, such as silicone oil from syringes, can serve as nucleation points for aggregation. Moreover, The high concentrations of protein often required for therapeutic effectiveness increase the likelihood of molecules associating with each other, further promoting aggregation. Denaturation is the process in which a protein loses its natural three-dimensional structure. This change is usually irreversible and leads to the complete loss of the protein's biological function. Various environmental stressors can induce denaturation, especially extreme temperatures and pH levels. These conditions can easily disrupt the weak non-covalent bonds that stabilizing the protein's folded shape. Proteins can undergo denaturation due to heat or cold. Thermal denaturation, caused by high temperatures, typically occurs between 40 and 80 degrees Celsius. Cold denaturation, which is less intuitive, can happen at temperatures above freezing, around 0 degrees Celsius. This process is driven by changes in how water molecules interact with the nonpolar regions of the protein. Therapeutic proteins tend to adhere to or bind with various surfaces they come into contact with. These surfaces include storage container materials such as glass and plastic, components used during manufacturing like purification media, and, importantly, air-liquid interfaces. Air-liquid interfaces are easily formed during routine handling procedures that involve agitation, such as swirling, shaking, or pumping. When proteins adhere to these interfaces, they can undergo conformational changes, which may compromise their structural integrity. This structural disruption can lead to a loss of the protein's active form, reducing its therapeutic effectiveness. Additionally, such conformational changes can serve as nucleation points that promote further protein aggregation, potentially impacting the safety and efficacy of the therapeutic product. Chemical degradation involves the modification of a protein's covalent structure through the formation or breaking of chemical bonds. These modifications can significantly affect the protein's function, stability, and safety. One common pathway of chemical degradation is oxidation. This process can influence therapeutic proteins at any stage of their life cycle, from production and purification to long-term storage. Oxidation is often caused by exposure to reactive oxygen species, which are generated by factors such as exposure to light, the presence of trace metal ion contaminants like iron and copper, or impurities within formulation excipients. Proteins contain amino acid residues that are particularly susceptible to oxidation. These include methionine and tryptophan, while cysteine residues are also highly prone to modification. The effects of oxidation can be severe, leading to changes in the protein structure, the formation of aggregations, and the inhibition or destruction of biological activity. Additionally, oxidation can alter pharmacokinetic parameters, potentially making the drug less effective or unsafe. Deamidation is a common chemical modification that primarily affects the amino acids asparagine and, to a lesser extent, glutamine. This process involves the conversion of the neutral side chain of asparagine into the negatively charged side chain of aspartic acid or isoaspartic acid, resulting in an additional negative charge on the protein. Such modifications contribute to the formation of what are known as acidic charge variants. The rate at which deamidation occurs is highly dependent on the local sequence context within the protein, as well as environmental factors such as pH and temperature. Additionally, isomerization of aspartate residues into isoaspartate can occur, which may alter the peptide backbone and potentially affect the protein structure and function. Hydrolysis is a chemical process that involves breaking down molecules by cleaving peptide bonds in the protein's backbone. This process results in the fragmentation of the entire molecule, which can compromise its structural integrity. For example, hydrolysis can cause the rupture of viral vector capsids or lead to the fragmentation of monoclonal antibodies. The process can be catalyzed by residual proteases that remain from the manufacturing process. Additionally, hydrolysis can occur spontaneously under certain conditions of pH and temperature, further increasing the risk of degradation. Overall, understanding the conditions that lead to hydrolysis is important for maintaining the stability and effectiveness of therapeutic proteins. The various degradation pathways are not separate processes, but are often closely connected. A single environmental stressor can set off a chain of events. For example, a slight increase in temperature or a change in pH can cause a protein to partially unfold, denature. This initial shape change can reveal hydrophobic amino acid residues that were once hidden inside the protein's core. These exposed areas can then stick together, leading to the formation of aggregates. Meanwhile, the changed shape might make chemically fragile residues, such as asparagine or methionine, more accessible to the solvent, increasing their risk of deamidation or oxidation. Oxidation itself is known to cause additional structural modifications that can make a protein more likely to aggregate. This shows that a control strategy cannot focus on a single degradation pathway in isolation. Managing aggregation, for example, requires a comprehensive understanding and regulation of the upstream physical and chemical events that start it. Furthermore, the specific environmental context determines which pathway of degradation is most likely to dominate. For instance, an acidic pH might be used intentionally to reduce the reactivity of certain residues and slow down oxidation processes. However, the same acidic formulation can significantly increase the risk of drug aggregation if it is diluted in dextrose and administered intravenously. This is due to a phenomenon known as isoelectric precipitation that occurs at the blood intravenous interface. Exposure to light specifically accelerates oxidative pathways, especially affecting tryptophan and methionine residues. Additionally, freeze-thaw cycles introduce unique stresses such as cryoconcentration of solutes and the formation of ice-liquid interfaces. These conditions promote aggregation and surface adsorption in ways that simple cold storage does not. This dependence on environmental conditions underscores the importance of conducting comprehensive force degradation studies. Such studies deliberately expose the drug to various stressors, including heat, light, extreme pH levels, and oxidation. The goal is to systematically identify the drug's vulnerabilities and determine the most relevant pathways of degradation. To address the inherent fragility of biologic drugs, the biopharmaceutical industry and regulatory agencies have adopted a modern approach based on scientific principles and risk assessment, known as quality by design. This framework emphasizes understanding and controlling the manufacturing process to ensure product quality. Central to this approach is the concept of the critical quality attribute, which acts as a vital link between the molecular characteristics of the drug, the manufacturing process involved, and the drug's final performance in the patient. The International Council for Harmonization's Guideline on Quality Pharmaceutical Development describes a critical quality attribute as a physical, chemical, biological, or microbiological property or characteristic that must be maintained within specified limits, ranges, or distributions to ensure the desired quality of a product. The quality by design approach signifies a fundamental shift from traditional quality control methods, which primarily depend on testing the final product, to a proactive strategy that incorporates quality into the product and process from the outset. The development process begins with establishing a quality target product profile, which outlines the intended characteristics of the final drug based on its clinical application. From this profile, a list of potential critical quality attributes is identified, representing all the attributes that might influence the product's quality. The subsequent step involves a systematic risk assessment guided by quality risk management principles to identify which of these potential critical quality attributes are genuinely critical. A team comprising experts from various disciplines evaluates each potential critical attribute based on its possible impact on the safety, efficacy, pharmacokinetics, and immunogenicity of the product, as well as the uncertainty associated with that impact. This risk-ranking process is iterative. As more information about the drug is gathered during its development, the list of critical quality attributes is refined and updated. It is important to note that this assessment should concentrate solely on patient safety and should not be influenced by the manufacturing process's current capabilities. Once the critical quality attributes are identified, the next step is to develop a manufacturing control strategy. According to the International Council for Harmonization's Guideline Q11, a control strategy is a planned set of controls derived from a thorough understanding of the product and process. Its primary purpose is to ensure that the process consistently performs as intended and that the final product maintains its quality. This strategy establishes important links between the critical quality attributes of the final product, the critical process parameters of the manufacturing process, and the attributes of the raw materials used. The development of a control strategy can follow a traditional approach. This approach relies on fixed set points, narrow operating ranges, and emphasizes extensive testing of the final product to ensure consistency. Alternatively, an enhanced quality by design approach uses a deeper scientific understanding of the process. This allows for the creation of a more flexible and robust control strategy. Such an understanding forms the basis for gaining greater regulatory flexibility. Two key concepts enable this enhanced approach. The first is process analytical technology, which is a framework for designing and controlling manufacturing processes through timely measurements of critical attributes during production. By employing sensors that operate in-line or at-line, along with multivariate data analysis, this method allows for real-time monitoring and control. This shift helps ensure quality is built into the process from the beginning, rather than being tested only at the end of production. The second concept is the design space. As defined in the International Conference on Harmonization's guidelines, a design space is a multidimensional range of input variables, such as material attributes and process parameters that have been scientifically demonstrated to ensure product quality. This operational window is established through experimentation, such as the design of experiments. Importantly, operating within this pre-approved range is not considered a regulatory change, providing manufacturers with significant operational flexibility. This flexibility supports continuous improvement without the need for constant regulatory submissions. The quality by design paradigm, enabled by critical quality attributes, process analytical technology, and the design space, fundamentally changes the relationship between manufacturers and regulatory agencies, such as the Food and Drug Administration in the United States and the European Medicines Agency. Traditionally, the approach focused on demonstrating that a fixed, well-defined process consistently produces a product that meets specified standards. Any modifications to the process, even minor ones, usually required a formal and often complex regulatory submission. In contrast, the enhanced quality by design approach involves gaining a thorough understanding of how and why the process operates. This includes understanding how process parameters and material attributes influence the final product's critical quality attributes. This comprehensive understanding enables manufacturers to define and secure regulatory approval for a defined operational space. Such approval confirms that any combination of parameters within this space will produce a product of the desired quality. This approval grants manufacturers greater flexibility to make process adjustments and pursue continuous improvements within the approved parameters. Consequently, the regulatory discussion shifts from mere strict process adherence to a focus on scientifically justified process control, fostering innovation and efficiency while maintaining product quality. The successful implementation of a quality by design framework depends heavily on having a comprehensive set of analytical methods. These methods must be capable of measuring critical quality attributes with high accuracy and precision. Selection of the appropriate analytical techniques should be customized to suit the particular molecule and its stage of development. Additionally, these methods need to be properly validated in accordance with guidelines established by the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. Common analytical techniques for evaluating critical quality attributes include various methods tailored to specific attributes. For size variants such as aggregates, size exclusion chromatography is the primary method used to quantify higher molecular weight species. When analyzing charge variants, a combination of techniques is employed to characterize charge heterogeneity, including ion exchange chromatography and capillary-based methods like capillary isoelectric focusing and imaged capillary isoelectric focusing. To examine the primary structure of the protein and post-translational modifications, peptide mapping is utilized. This process involves enzymatic digestion of the protein followed by liquid chromatography mass spectrometry, which serves as the gold standard for confirming the amino acid sequence and identifying the location and nature of modifications, such as oxidation or deamidation. Glycosylation, being highly complex, requires a multifaceted approach. It involves analyzing released glycans, glycopeptides, or the intact glycoprotein. Techniques used include hydrophilic interaction liquid chromatography, capillary electrophoresis, and various advanced mass spectrometry methods to characterize the diverse glycan structures. While this analytical toolkit is powerful, it also presents a significant bottleneck in fully realizing the vision of quality by design. The goal of quality by design, especially the implementation of process analytical technology for real-time process control, pushes the limits of current analytical capabilities. For example, identifying critical quality attributes requires highly sensitive methods capable of detecting and quantifying low-level variance. Establishing a robust design space depends on generating extensive amounts of high-quality analytical data through comprehensive experimentation. A true process analytical technology-based control strategy requires sensors that can be placed inline or at the side of the process, providing real-time feedback on critical quality attributes or their surrogates. However, current technologies have limitations. Traditional techniques for analyzing charge variance, such as ion exchange methods, use high-salt buffers that are incompatible with direct mass spectrometry analysis. This incompatibility hinders the ability to identify variants in real time as they are separated. Similarly, analyzing complex glycosylation patterns on intact proteins in real time remains a significant challenge. Therefore, advancements in analytical science, particularly in rapid mass spectrometry-based techniques and robust process sensors, are not merely academic pursuits. They are essential prerequisites for fully leveraging the modern regulatory framework and enabling the practical application of advanced process control. The ultimate goal of identifying and controlling critical quality attributes is to ensure that any variability between batches in the manufacturing process does not lead to clinically significant differences in safety or effectiveness for patients. Achieving this goal requires establishing a clear and evidence-based connection between specific molecular characteristics and their effects on clinical performance. Protein aggregation is commonly regarded as a significant critical quality attribute because of its strong potential to increase the immunogenicity of a therapeutic protein. The formation of protein aggregates can disrupt the body's immune tolerance to the therapeutic protein, which may trigger the production of anti-drug antibodies. Aggregates tend to be more immunogenic than monomeric proteins because their particulate nature and repetitive epitopes enhance their uptake by antigen-presenting cells. This improved uptake can lead to a more robust activation of both the innate and adaptive immune responses. The clinical consequences of an anti-drug antibody response can be severe. These antibodies can bind to the therapeutic protein and accelerate its clearance from the body, which neutralizes its effect and diminishes its efficacy. In the most dangerous cases, these antibodies can cross-react with a patient's own endogenous version of the protein, leading to serious and sometimes life-threatening adverse events. A well-documented clinical example illustrating this risk is the increased occurrence of pure red cell aplasia, a severe form of anemia, in patients treated with a specific formulation of recombinant human erythropoietin. Investigations have linked this adverse event to the development of neutralizing antibodies that targeted not only the drug, but also the patient's native erythropoietin. The formation of protein aggregates, potentially induced by leachates from container stoppers, has been identified as a key contributing factor. Similar links between protein aggregation and immunogenicity have been observed in clinical studies involving other biologic therapies, including interferon beta and human growth hormone. The type and size of these aggregates are also critical factors. Higher molecular weight aggregates and particles in the subvisible range, specifically between 0.1 and 10 microns, are considered particularly potent in provoking an immune response. Glycosylation is an enzymatic process that involves attaching sugar chains, known as glycans, to a protein. This post-translational modification is crucial for many biological medicines, especially monoclonal antibodies. The specific pattern of glycosylation can greatly influence the drug's stability, effectiveness, pharmacokinetics, and safety. Because of this, glycosylation is considered a critical quality attribute of these biologics. The impact of glycosylation heterogeneity on clinical performance is complex and multifaceted. One significant aspect is its effect on the efficacy of therapeutic antibodies. For many cancer-fighting monoclonal antibodies, a primary method of action involves recruiting the immune system to destroy cancer cells through a process called antibody-dependent cell-mediated cytotoxicity. In this process, the antibody marks the cancer cell for destruction by immune cells such as natural killer cells. This interaction is facilitated by the antibody's Fc region binding to Fc gamma receptors on the natural killer cells. The absence of a specific sugar, called core fucose, on the Fc glycan, known as a fucosylation, has been shown to dramatically increase this binding affinity. As a result, the antibody's ability to induce cell destruction through this mechanism is significantly enhanced. Conversely, the presence of terminal galactose on the glycan can improve another immune response by strengthening the binding of a complement protein, leading to increased complement-dependent cytotoxicity. This demonstrates how different glycosylation patterns influence the various pathways through which therapeutic antibodies exert their effects. The half-life of a biologic drug in the bloodstream is an important factor in determining how frequently it needs to be administered. Glycosylation, the process of adding sugar molecules to proteins, plays a significant role in this aspect. When terminal sialic acid residues are present on the glycans, they can extend the drug's duration in the body. This is because sialic acid can mask underlying galactose residues, which, if exposed, would be recognized by receptors in the liver known as asialoglycoprotein receptors. Recognition by these receptors leads to rapid clearance of the drug from circulation. Conversely, if the glycan structures are predominantly of the high mannose type, they tend to be cleared more quickly through receptors that specifically recognize mannose residues. This accelerated clearance results in a shorter half-life for the drug. The immune system is trained to recognize human glycan structures. When the manufacturing process results in the production of non-human glycans, such as N-glycolylneuramic acid or galactose alpha-1, 3, galactose, these substances can be perceived as foreign by the immune system. As a consequence, they may trigger an immune response. Therapeutic proteins, particularly monoclonal antibodies, are composed of a diverse mixture of molecules that differ slightly in their overall surface charge. This variation, known as charge heterogeneity, is considered a critical quality attribute because these different variants may exhibit distinct stability, potency, and pharmacokinetic profiles. The acidic variants of these proteins often result from chemical modifications such as deamidation of asparagine residues or the addition of sialic acid-containing glycans. Conversely, the basic variants can occur due to incomplete processing during manufacturing. For example, when there is a failure to remove lysine residues from the ends of the antibody heavy chains. The clinical impact of charge variants is greatly influenced by where and how the modifications occur. Changes within the regions of the antibody responsible for binding to the target, known as complementarity determining regions, can directly affect the drug's effectiveness by altering its ability to attach to its target. On the other hand, modifications in the fragment crystallizable region are more likely to influence the antibody's effector functions, which are responsible for recruiting other components of the immune system. The overall charge of a protein can significantly influence its pharmacokinetics through nonspecific electrostatic interactions with negatively charged surfaces of cells and tissues. Studies conducted in preclinical settings have demonstrated that increasing the positive charge of an antibody, which is indicated by a higher basic isoelectric point, can result in faster clearance from the bloodstream and greater retention within tissues. Additionally, variants of the protein with different charges may exhibit varying levels of binding to the neonatal Fc receptor, a receptor responsible for recycling antibodies and thereby extending their half-life in the bloodstream. Some variants with increased basic charge have shown enhanced binding to this receptor, which could potentially lead to a longer duration of presence in the circulatory system. The study of critical quality attributes reveals that variability is not always harmful. Although the primary goal of manufacturing is to ensure consistency by reducing unintended differences between production batches, a deeper understanding of the relationships between structure and function enables the deliberate engineering of specific profiles of critical quality attributes to develop therapeutics that are more effective than existing ones, known as biobetter drugs, with improved clinical properties. For example, Removing core fucose from an antibody used in cancer treatment is not a sign of degradation. Rather, it is a purposeful modification aimed at enhancing its antibody-dependent cellular cytotoxicity activity, which results in better cell-killing capabilities. Similarly, glycoengineering to increase the level of sialylation can prolong a protein's half-life, potentially allowing for less frequent dosing and greater convenience for patients. This demonstrates that the ultimate objective of managing critical quality attributes is not merely to prevent unwanted variability, but to understand and control all sources of variability. Leveraging this knowledge allows for the design of superior, next-generation drugs. Developing biosimilars presents an immense challenge. A biosimilar must be highly similar to an approved reference product, with no clinically meaningful differences. Achieving this requires more than simply matching a few key critical quality attributes. It involves replicating the complex, interdependent molecular fingerprint of the original product across a wide range of attributes. The clinical significance of any variation in a single critical quality attribute is highly dependent on the context. For example, enhanced antibody-dependent cell-mediated cytotoxicity is beneficial in cancer therapy, but could be harmful when used in an immunosuppressive drug. Similarly, a small amount of protein aggregation might be clinically irrelevant for some products, but could prove catastrophic for others, such as a product designed to replace a deficient endogenous protein. Because these critical quality attributes are often interconnected, such as how glycosylation influences the charge profile or how oxidation can lead to aggregation, their clinical impact cannot be assessed in isolation. Preserving the integrity of a biological therapeutic product requires a comprehensive approach that covers both the manufacturing environment and the setting where the treatment is administered to patients. This approach involves developing a robust formulation, practicing strict aseptic compounding procedures, and ensuring careful management of cold chain logistics from production to clinical use. The formulation serves as the first line of defense against the degradation of the therapeutic protein. A well-designed formulation creates a microenvironment that effectively stabilizes the protein, protecting it from various stresses that could compromise its effectiveness and safety. The formulation composition involves the careful selection of key components to maintain the stability of the biological product. It is essential to keep the pH at an optimal level to ensure both conformational stability, which relates to the protein structure, and colloidal stability, related to its solubility. This balance is achieved through the use of specific buffer systems. The ionic strength of the formulation, which influences the surface charges of the proteins, is controlled by adjusting the salt concentration to help minimize aggregation. Stabilizing excipients are added to protect the protein during processing, storage, and handling. These include sugars such as sucrose, amino acids like arginine, and surfactants such as polysorbates. However, it is important to be cautious, as some excipients can introduce destabilizing impurities. For example, peroxides may be present in polyethylene glycol, and free fatty acids can be found in polysorbates, both of which can actively degrade the protein. Environmental stressors are a critical consideration beyond formulation. Strict control over the external environment is essential because biologic products are highly sensitive to temperature excursions outside their specified storage range, which is typically between 2 and 8 degrees Celsius. Any deviations from this range can lead to irreversible degradation and a loss of efficacy. Freeze-thaw cycles are especially damaging, as the formation of ice crystals and the cryoconcentration of solutes can disturb protein structures and promote aggregation. Additionally, exposure to ultraviolet and visible light can cause photooxidation and fragmentation of the biologic molecules. Mechanical agitation from shaking or pumping can also be harmful, as it may cause denaturation at the interfaces between air and liquid. Overall, maintaining optimal environmental conditions is vital to ensure the stability and effectiveness of biologic therapies. The process of preparing a biological product for patient administration, especially when used for intravenous infusion, is considered high risk and involves many potential points where errors can occur. To minimize these risks, the process is strictly regulated by rigorous standards designed to ensure patient safety. These standards are primarily detailed in the United States Pharmacopoeia's general chapters related to sterile preparations and hazardous drugs. Additionally, best practice guidelines from organizations such as the Institute for Safe Medication Practices are also followed to maintain high safety and quality levels. Aseptic processing is essential to prevent microbial contamination during sterile compounding. This process must take place in a highly controlled environment to ensure safety and sterility. Typically, a primary engineering control device, such as a laminar airflow workbench or a biological safety cabinet, is used. This device provides an environment with air quality classified as International Organization for Standardization Class 5. Such a primary engineering control is usually housed within a buffer area that meets the International Organization for Standardization Class 7 standards, commonly referred to as a cleanroom. Personnel involved in the process are required to adhere to strict procedures for hand hygiene and garment preparation. Additionally, all surfaces and components must be thoroughly decontaminated before they are brought into the sterile area. Handling hazardous biologics requires careful precautions, especially when dealing with many modern biologic drugs, such as antibody-drug conjugates and certain monoclonal antibodies used in cancer treatment, which are classified as hazardous drugs. The United States Pharmacopeia calls for additional protective measures to prevent occupational exposure for healthcare workers. These measures include the use of specific containment engineering controls, such as a compounding aseptic containment isolator and appropriate personal protective equipment. Specialized handling procedures are also essential. One key engineering control recommended is the use of closed-system drug transfer devices, which help minimize the generation of aerosols and prevent leakage during drug transfer. This not only protects the handler, but also maintains the sterility of the product. Reconstitution and dilution are important steps when handling many biological medicines. These medicines are often supplied as powders that have been freeze-dried. Before use, they need to be reconstituted with a sterile liquid. This process must be carried out carefully, using gentle agitation to dissolve the powder. It is important to avoid causing foaming or shear-induced damage to the medicine during this step. After the medicine has been reconstituted, it is usually further diluted in a large volume intravenous fluid, such as an intravenous bag. This dilution is a critical step because it significantly decreases the concentration of stabilizing substances that were present in the original formulation. As a result, the protein may become more susceptible to breaking down or sticking to surfaces within the intravenous bag. Intravenous admixture compatibility is a complex subject that requires careful consideration of the diluent and infusion materials used. The selection must be based on specific compatibility data related to the product. For example, many monoclonal antibodies formulated at an acidic pH are incompatible with dextrose 5% in water. Diluting these medications in dextrose solution can cause rapid protein aggregation, which is believed to occur due to isoelectric precipitation when the low ionic strength, acidic solution contacts blood at the infusion site. Therefore, normal saline, which is a sodium chloride solution at a concentration of 0.9%, is typically the preferred diluent for most monoclonal antibodies. Additionally, the biologic medication can adsorb onto the plastic surfaces of intravenous bags made from materials such as polyvinyl chloride or polyolefin, as well as tubing, syringes, and inline filters. This adsorption can lead to a significant loss of the intended dose. To prevent these issues, compatibility studies should be conducted to ensure that there are no adverse interactions between the biologic and contact materials. These studies also help verify that no harmful substances, known as leachables, are released from the materials into the medication. Ensuring compatibility and safety is essential for the effective and safe administration of biologic therapies. The final stages of a biological product's journey, reconstitution, dilution, and administration, represent a crucial transition from the highly controlled pharmaceutical supply chain to the more variable clinical environment. This shift introduces numerous new variables, including a different solvent system, extensive contact surfaces, significantly altered concentrations, and new sources of mechanical stress. A manufacturer's control strategy cannot end at the pharmacy door. It must extend into the clinical setting by providing robust, validated, and clearly understandable instructions for use. Therefore, Comprehensive in-use stability and material compatibility studies are not optional additions, but essential extensions of the product's characterization. These studies effectively define a design space for safe clinical handling and administration. Maintaining the integrity of a biological product from the manufacturing site to the patient requires an unbroken, temperature-controlled supply chain. This supply chain is commonly referred to as the cold chain. It involves complex logistical operations that are regulated by strict guidelines and standards, such as current good manufacturing practices and good distribution practices. These regulations ensure that the biological product remains safe, effective, and of high quality throughout the delivery process. Most biologic medicines need to be stored at temperatures between 2 degrees Celsius and 8 degrees Celsius. Some advanced therapies, such as cell and gene therapies, might require freezing at -20 degrees Celsius or colder, including cryogenic conditions below -150 degrees Celsius. Maintaining these temperatures is achieved through specialized temperature-controlled packaging systems. These systems feature high-performance insulated containers, carefully selected refrigerants like gel packs, phase-change materials, or dry ice, and temperature monitoring devices. To ensure reliability, the entire packaging must undergo rigorous validation testing to confirm it can maintain the required temperature throughout the entire transportation process, even in worst-case external temperature conditions. Monitoring and data logging. The modern cold chain has developed significantly from a passive approach, where shipping depended on luck and hope, into an active, data-driven system that emphasizes constant monitoring and quick response. The use of electronic data loggers and real-time sensors connected through the Internet of Things has become standard practice in the industry. These devices continuously record the temperature history of shipments, creating an auditable trail that is crucial for meeting regulatory requirements. More advanced systems offer real-time alerts, allowing logistics providers to take proactive measures, such as re-icing delayed shipments, to prevent temperature excursions that could damage the products. This evolution has transformed the cold chain from merely a physical process of transporting goods into a system where data integrity is vital. The quality of the final product is now closely linked to the accuracy and reliability of the data collected during transit. The last-mile challenge, the final stage of distribution, from a central pharmacy or distribution hub to the clinic or patient's home, is widely recognized as the most vulnerable part of the cold chain. This last mile is subject to numerous risks, including unpredictable traffic and weather delays, diverse climatic conditions, and less controlled handling environments. Mitigating these risks requires extremely robust packaging solutions, detailed contingency planning for potential delays, and clear communication with the end user to ensure proper handling upon receipt. Ensuring the consistent clinical performance of a biologic therapeutic is a complex undertaking that demands a holistic, life cycle-wide approach to quality and risk management. It requires the integration of deep scientific understanding with rigorous operational control, recognizing that the journey from cell culture to patient administration is a single, interconnected process. The ultimate goal is not the impossible task of eliminating all variability, but the achievable and essential goal of understanding, controlling, and managing variability to ensure it remains within a clinically irrelevant range. A holistic approach to risk management is fundamental in biologics manufacturing, rooted in the principle that the process itself is the product. This concept recognizes that even minor alterations in the process can significantly impact the final molecule. It is essential to extend this principle beyond the confines of the manufacturing facility to include the entire supply and administration chain. A successful control strategy should not be isolated within a single department, but must instead be an integrated framework. This framework should draw upon knowledge from various scientific and logistical disciplines. For instance, formulation science involves selecting optimal pH levels, buffers, and excipients to stabilize the protein against anticipated stresses. Process development focuses on understanding how critical process parameters in both upstream and downstream manufacturing influence the critical quality attributes of the final drug substance. Analytical science plays a crucial role in developing and validating robust, phase-appropriate analytical methods to accurately monitor all identified critical quality attributes. Additionally, logistics and supply chain management are vital to implementing a qualified and continuously monitored cold chain, which protects the product from environmental stresses during transit. Finally, clinical pharmacy practice involves providing clear and validated instructions and understanding the risks associated with reconstitution, dilution, and administration. These measures are essential to maintaining the drug's integrity up to the point of patient contact. Manufacturing processes for biologics are not static. They evolve throughout the product lifecycle due to scale-up, process improvements, or site transfers. Whenever such a change is made, regulatory agencies require a comparability exercise to demonstrate that the product made by the new process is highly similar to the product made by the old process, and that the change has not adversely impacted its quality, safety, or efficacy. These exercises are a critical application of the CQA framework. They rely on the comprehensive analytical toolkit developed for the product to perform a side-by-side comparison of all key CQAs. The goal is to provide a weight of evidence demonstrating that the CQA profiles of the pre- and post-change material are statistically equivalent and fall within the established, clinically justified acceptance criteria. A successful comparability exercise provides the scientific justification for foregoing additional, costly clinical trials, bridging the clinical data from the original process to the new one. The field of biopharmaceutical development continues to advance, driven by new technologies and evolving regulatory paradigms. The ongoing development of advanced analytical technologies, such as inline and online sensors for spectroscopy and mass spectrometry, is enhancing capabilities in the field. These improvements facilitate the realization of real-time release testing, a method that allows manufacturers to evaluate product quality continuously. As a result, manufacturing processes become more adaptive and efficient, ensuring consistent product quality throughout production. Predictive modeling and artificial intelligence are transforming the landscape of process development, manufacturing, and cold chain monitoring. The large volumes of data generated in these areas present an opportunity for analysis using advanced algorithms in machine learning and artificial intelligence. These powerful tools can be employed to construct models that predict potential process deviations, identify sources of variability, optimize shipping routes, and develop more adaptable and intelligent systems for risk management. Regulatory harmonization is an important aspect in the global pharmaceutical industry. International organizations, such as the International Conference on Harmonization, work to establish unified standards across countries. However, differences in how regional regulatory authorities interpret and implement these standards can still present challenges. These discrepancies can complicate efforts by companies to market a single product worldwide using a unified control strategy. Ultimately, the central challenge in biopharmaceutical development is the management of inherent variability. Unlike chemically synthesized small molecules, biologics are produced by living cells, which introduce a natural level of heterogeneity into the final product. No two batches of a biologic will ever be perfectly identical. Regulatory agencies like the FDA recognize and accept this inherent variability. The purpose of the entire QBD framework is not to eliminate this variability, but to deeply understand its sources and its impact. By establishing the link between molecular attributes, CQAs, and clinical outcomes, a manufacturer can define an acceptable range of variation for each CQA, the design space, within which the product is proven to be consistently safe and effective. The pinnacle of quality assurance for a biologic is therefore not the creation of an identical product every time, but the robust, data-driven demonstration that all batch-to-batch differences are understood, monitored, and controlled within a space that has no clinically meaningful consequence. This scientific narrative of controlled variability forms the core of a modern regulatory submission, such as a biologics license application, BLA, to the FDA, or a marketing authorization application, MAA, to the EMA. The submission is not merely a collection of disparate data, but a cohesive scientific argument. The manufacturing sections detail the control strategy, presenting the evidence for the manufacturer's understanding of CQAs and their link to CPPs. The clinical sections provide the safety and efficacy data that justify the acceptance criteria for those CQAs. In essence, the submission tells the story of how the manufacturer identified what was critical for patient outcomes, learned how to control it throughout the product's life cycle, and ultimately proved that this control results in a consistently safe and effective medicine. Mastering the myriad challenges of stability, compounding, and logistics is about building this robust, data-rich narrative to satisfy regulatory scrutiny and, most importantly, to safeguard the well-being of every patient. This was an episode of the Nexus Clinical Podcast. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.